All right. So we started with the first one. And again, we will mainly stop at some of the important slides. And the first one, we start with the cell culture. And here, some of the important parts is that what exactly we are doing right here. And there are two concepts that we are trying to learn here, that when you are starting a cell culture, your source could be two different things. That's the important part. And here we are just only showing one. So one is that you can get a live tissue. And after that, getting the live tissue, you can actually extract these cells and start a population of cell culture. So how does this work? You will take a piece of tissue and then mince it off and then take these connective tissues off and everything. And then usually you use these trypsin collagenase and the EDTA, and these will basically disrupt the cell adhesion molecules, because calcium is needed for the cell adhesion molecules to work. So if you take this EDTA, and it will just mop up or chelate the calcium, and that will disrupt the tissues or basically make the cells separate, and you can culture them. Now, what you can see that many of the tissues, remember that when you are talking about the tissue usually will comprise of just only one kind of cell. But if you do not carefully process your tissue, you may have a couple of different types of cells as well. And that may give you a heterogeneous population of cells. And there is a way to separate out the cells and just make a cell, basically cell cloning. We dilute the cells and then separate the cells out and culture the ones that you actually want. So this is known as the primary culture. What primary, again, remember that a primary culture means that you have started this culture from a live tissue, which is not cancerous or anything. It's just a live tissue from an embryo, from a fetus, or a part of an adult animal, human body, whatever it is, a live tissue, and started that is a primary cell culture. So when you do this primary cell culture, I'm skipping a couple of slides and we will come back. You are going to see that it will just grow over the period of time, and it will grow for a number of generations. Now, 50 is a lot of generations. You may see senescence before that. And after that, you are going to see that the cells are going to die. So 100, hardly they will go into that. It between 25 and 100 passages that we say, that basically you take the cells, you put it in a flask, and then grow it for 5 days. It will be all confluent all over the place. Then you again take those cells off and then passage it to a new flask and keep doing this and grow them and multiply them. You can just have many cells out of those. But all these cells, no matter how many times you passage those over the period of time, they will die because they are all mortal. So this is your primary cell culture. So some of the labs will use these kind of cells and doing that. But the majority of the labs which do cell culture experiment on a regular basis for most of the time, unless it is necessary, people do not use a primary cell culture because they are finite and you have to start back again from the scratch, right, like this, the first step after that. So what they use, what we usually call a cell line. So again, when we call a cell line, usually we mean, again, there is not a very strict wording or nomenclature with that. We can call a primary cell line or an immortal cell line. But usually when we use the word cell line, conventionally, it means that it is immortal and it is derived from uncancerous tissue and it will go forever. And it's derived from a tumor that undergoes spontaneous genetic change, which is oncogenic transformation. So what does that mean? That means that there are, in the cell, there are some cell cycle control proteins which actually control the cell division. And over the period of time when the cell divide, there will be accumulation of mutations, and the cell understands that after a certain time, that cell has accumulated enough mutations and it's not healthy to keep that cell alive, and it will automatically stop the cell from growing, and that's in essence, it will die. In these cell lines, no matter what mutation it accumulates, the cell cycle control protein has a mutation and it cannot stop growing. It will not allow the cell to die. So they are genetically not identical to live cells. And say from HeLa, HEK 293, this is a Chinese hamster ovary cells. SF9 is from an insect cells. And there are hundreds and hundreds of these different types of cell line that is commercially available. One lab gets it from the other. You can just get it from a frozen culture from the ATCC, which is called the American Type Culture Collection. They have the collection of all the available cell lines, and they will just send you over dry ice these, and you thaw it in the lab and start doing, and it will go from ever. If you have read the book, The Immortal Henrietta Lack, from where the HeLa was actually, so 1956, this African-American lady, she went to Johns Hopkins, and at that time, the cells were taken from her cervical tumor. And that was an HPV or human papilloma virus derived tumor. And there was lots of legal things at that time. Without asking her, it was taken. And it is probably one of the most common cell line that grows in tens and thousands of laboratories all throughout the world, and it will grow forever. So that's basically the cell line. Now, what happens that when you take these cell line, interestingly, the mouse cells, on the other hand, although they are mammals, they are not primates. And you can see that in between the mammals and mammals, also primate and non-primate, how things actually vary. If you take some cells, which are human cells, and you start a primary culture like this, what you are going to see that after some time, they are going to die. This is always, it's going to happen. But if you take the mouse cells, sometimes, and again, just make sure that you understand that sometimes they will potentially die after, which is a senescence. But what happens sometimes, not always, remember that, just by chance, if you're lucky, sometimes you may see that all the cells are dead and there are some few colonies of cells, they are started growing back up. And this is an emergence of an immortal variant cell line. So what is going on here in the mouse cells that the human cells or the primate cells didn't do? So just imagine, just like within the mammals even, the mouse actually do not have a very robust anti-cancer property, so that some of the cells which does have the mutation of the cell cycle, if you have a mutation in the cell cycle, essentially right here, there are lots of other proteins that will kill the cell and not allow it to grow. So these are our robust anti-cancer property. We have to have 3 to 7 mutations, at least 3 to 7 mutation in the same cell before they can become cancerous. And that's why it is still rare, like comparatively rare. Mouse doesn't have that control that robust. That's why they can give rise to this immortal variant cell line in culture. But you will not observe this immortal cell line from a primary culture in the human cell line. They will always die. So again, take home message, you have a primary cell line that you can derive from the tissue. It is mortal. You can do experiments with that particular tissue or the cells for a certain period of time. But if you are doing a long-term, any experiment just where you need a particular cell line works with that type of viruses, you just have a cell line that just allows the virus to grow, and you can just do that. So these kind of experiments that you can actually do from these cell lines, which are immortal. Now, let's go back to a couple of different things that what you require from here. So to do this cell culture, you need a conditioned pH ionic strength. Ionic strength means like you need a lot of different, like a sodium, then potassium, you need fluoride, then you need carbonate, bicarbonate, all these different ions, hydroxyl, all these different electrolytes that we essentially what we tell that we need those ionic strengths. Essential amino acid, vitamins, peptide growth factor. And now these are often supplied from the serum, fetal calf serum, some exceptions. But now the scientists have developed many media which are serum-free media, because serum is an extremely expensive part of the cell culture and increases the cost. But you can actually make these peptide growth factors now artificially, and you can grow many cells in a serum-free medium. Antibiotic, antimycotic. Antibiotic is against the bacterial growth, and antimycotic is antifungal. Just remember that the bacteria and fungus both grow exponentially, much, much faster than the animal cells, which usually divide in about 24 hours. So if there is a contamination, even a slightest, just one contamination of a bacteria or a fungus, they will take over and your cell will be contaminated. So you need antibiotic, antimycotic. On top of that, you absolutely need to handle everything in a sterile condition inside a laminar airflow hood or those safety cabinets that you can see in some of those pictures and supplementary diagram as well. Many cells actually need a negatively charged solid surface that mimics the extracellular interaction in the cells. And you really do not have to do very special thing for that. All these cell culture Petri plates or flasks and dishes, they are usually made of polystyrene plastic. And when they are made, their surface where the cells are growing basically etched a little bit. It's just the manufacturing process. And these plastics are negatively charged. These will interact with the extracellular interaction of the animal cell. So now the important part is that the cells like to attach to each other. And these attachments are usually by the cell adhesion molecule. So this CAM, so just if you're just in cell adhesion molecule, definitely underline that. And some of these are collagen, fibronectin, laminin. And these are some of the proteins we are going to talk about in very detail and their functioning in the later part when we talk about the cellular interaction. These proteins in general, all of these cell adhesion molecules need calcium for their function. And if you see that right here, you need an ionic strength. So usually what we have that in one of the conditions, what we give them is calcium chloride. So calcium chloride is one of the component of these cells. And what you have there is Ca2 plus and 2Cl minus calcium chloride. So CaCl2. So this is one of the component, and this calcium chloride gives you the functioning of the collagen, fibronectin, and the laminin. So this is a part of this ionic strength, and calcium chloride provides that. Now, what you do, that this is the interesting part, that if you just go back to the previous slide, see that right here. So right here is the EDTA. This is a disrupting agent, along with the trypsin and the collagen is. So what the trypsin and the collagen is doing, these are proteases, or you can call them proteinases, which disrupt the proteins. So the proteinases are going to disrupt or partially digest these cell adhesion molecules, which are on the plasma membrane of the cell. On the other hand, this EDTA, which is the calcium chelator, you have the calcium chlorides right here. You have the Ca2 plus right there. These Ca2 plus, these are going to be mopped up by the EDTA. So this will be chelated by the EDTA, and you do not have the calcium available for the collagen, fibronectin, laminin to function, or that is inactivated by the calcium removal. So this actually helps the cells to come apart. So what we mean by this? So when we are culturing the cells at the very beginning, right here, we need to separate the cells. But when we are also growing the cells, remember that is also a very important concept. When we're growing the cells are attached to each other, and this is at the bottom of the Petri plate. So suppose if it's a Petri plate or a flask, the cells are growing like this. So now, after a few days, they are just all over the place. We call this as a confluence, or confluent, 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 or confluence means that they are all over the place like a lawn. And now I need to passage them. We need to move it to a new one. So you just put media on top of that. And now if you want to take these cells out, they're attached to each other again by their cell adhesion molecule. And there is calcium chloride in the medium, and you can scrape them and they will get injured, they will die. So what you do, that again, you will use, now this is not your tissue, this is your already these heterogeneous, or it could be again homogeneous depending upon the tissue, homogeneous population of cells in a flask. And you will again use a protease EDTA, you will add in here. So what will happen, that they will disrupt these cell adhesion molecules, which are attaching them with the cells as well as the substrate. So they will all come loose. They will just start floating just within 5 minutes. You incubate with them about 5 minutes, they will start floating in there. And all you do is you have to take those and you have to just spin them down, remove this trypsin EDTA, this material, so that they are not in the solution, and you just dissolve the pellet or the cells very gently with the phosphate buffer saline or the media, and you put a few cells right here. So it will start with a few cells. They will attach to the bottom within a few hours. And this gentle treatment with the protease actually do not kill the cells. It is just partially digest, and they will express those protein again, and they will attach, and then they will divide, and they will again become confluent within a few days. And then you take those cells again, do the same treatment, and put it back in there, and you can just multiply them, and your cells will be growing this way. So this is how you basically do the cell culture. So that's about pretty much all of it that you need to know from here, that how you culture the cells. Now, I go a little bit of more detail in here as well, because we do not have a lab component. So one of the things that is important take-home message for your exams, that know the function that what trypsin collagenase is doing. Remember that they are basically inactivating the collagen fibrinogen laminin, which is known as the cell adhesion molecules. What EDTA is doing, they are chelating the calcium, which is available from the calcium chloride, say for example, in the media. And the removal of calcium will also inactivate these, and you will be able to disrupt the cells, or they will be able to come off the media. Remember that you need those from the very start of the primary culture, but when you are maintaining the cells in the flask, you also need to use the protease or EDTA, trypsin EDTA, to passage the cells from one flask to the other, and so on. So I will spend a good amount of time on that. Sometimes people ask is we are looking at this embryonic cell need additional growth requirements, and do you need to remember those? Don't worry about those. And again, remember that these things will be accessible to you. So something that if you need to look up in between the lines, you can always look up for your exam. It's just that you can conceptually answer the questions that you may not find. Those are the ones that we are going to point out directly. But again, do not have to memorize these, but just shows that. So this is just basically what shows, essentially what it basically showing, that if you're culturing these embryonic cells, the cell which are at the bottom, which are these darker color, these cells are confluent, and these are called the feeder cells. And they secrete some of these growth factors to support the cells, which are those embryonic stem cells, which are growing at the top. So if you just want to ask me that what is confluency, this bottom cells, this is confluent. So you have to take them off and just put it in a new cell. But again, no direct question. All right. So now what you have, suppose you have a primary tissue, like you have a primary tissue and you have collected some of the cells, heterogeneous population, and you need to isolate just only one type of cell. Say, for example, you take a spleen and you want a B cell or a T cell. It's very specific for a particular purpose. How there could be all other different types of tissue in the or cells in the tissue. Now you want to separate them. So this is what you will do, that we can separate them on physical property, physiological property by flow cytometry. So these are different techniques from heterogeneous population of cells, that how you make the homogeneous population of cells. One of them is the flow cytometry that we are talking about. So suppose this is just an example. All cells essentially have some signature protein, and we have a name for them. We call them as marker proteins. So these marker proteins are cell surface proteins for a particular cell type. Like say, for example, we do have 200 different cell types in a human body. And each one of the cell, there will be at least one or few proteins which make that cell that cell. So these are the marker proteins. And otherwise, how your body actually understand that this type of epithelial cell is an epithelial cell. This is a liver cell. Of course, there will be all different other proteins inside the cell, which is different. But these marker proteins are usually cell surface proteins, which allows your body to understand what kind of cell it is. So this marker protein for the T cell is, say, for an example, this CD3. So CD3 is one of them. And this Thai 1.2 is another marker for this. And both of these marker proteins are expressed on the T cells, and these are the T lymphocytes. So if you take, say, for an example, a spleen from a mouse or a human being, you will have lots of these T lymphocyte or the T cell and the B cell and other cells, a mixture of a lot of different cells in there. So how you separate them? By the help of flow cytometry. So for that, you need some reagent, and these are commercially available. One is anti-CD3 antibody. What it means that it is an antibody which recognizes the CD3. So this is CD3. This is your black one is an Y-shaped antibody molecule, and there is a red fluorescence conjugated to it. So it's a red fluorescence conjugated anti, means against CD3 antibody. This will recognize the CD3. And the same way, you have a green fluorescence conjugated anti-Thai 1.2 antibody. So you have two things. You have a red and a green. And these other immune cell systems of spleen cells, they do not have these markers, and nothing will bind to them, as you see. So when you homogenize the spleen, and if you mix these antibodies, you can see these antibodies will specifically bind to the T cells. They will leave the other cells alone. And you can just take this property and run this cell through a flow cytometry to sort the T cells out. And how do you do this? So let's go to the next slide. So here in this process, it's a huge instrument. It's a multi-million-dollar instrument, very state-of-art technology. So in this instrument, you have this fine nozzle through here. You put the cell suspension where you have all the mixed cells, and there is basically called a sheath fluid, which basically is covering the cells, pass through a tiny nozzle right here. So one cell kind of can pass through each time. So one cell, just one cell. So this very tiny orifice in there through which it can pass. And you can see that there are these two different kinds of cells that you are seeing. And again, do not right here, you have this red and the green thing right here. And here, do not correlate the colors from this picture, fluorescent, non-fluorescent cells, with this. So just don't make this here, because this is made by me. This is your textbook picture. You have two different fluorescent type of cells. And you can see that one is a bright pink or reddish, one is a lighter pink. These are the fluorescent cells, which means that they have two different kinds of antibodies conjugated to something. One is dark red, one is light red. And then you have a non-fluorescent cells right here. And these are your fluorescent cell droplets. These are non-fluorescent cell droplets. So what is going on? You have all these cells being passed through here. So a laser beam is usually shined on top of this, and this will be detected by two different things. One is a fluorescent light detector. That will be the color based on the antibodies and the fluorophores that is conjugated to that. And another will be the scattered light detector. This will be dark or light. What it means that if there is a cell here, it will not allow this light to pass, and there will be a dark spot. It will count the cell that, okay, one cell is passing. If there is light going on there, that means that there is no cell. So it can count the cell that is going one at a time, and it can also count the fluorescence coming in there. So this is the double detection, what it means, that why the scattered detector is in there. It can just measure every cell that is going through here. What it does, that due to these two plates, depending upon the fluorescence that you have, they will put a slight charge on top of the cells by these plates. Now here it just shows that there are two different types of fluorescence. Now there could be an 8-channel fax, which can put like 8 different color cells at the same time, 8 different antibodies, and you can separate out 8 different cells from a tissue. So it's unbelievably amazing and complicated. So what if there's a non-fluorescent cell? It can still measure the number of cells that's passing through, but it will be collected through this channel where there will be no charge. It will fall straight through. Whereas what happens that if they put a charge in those, right here, there are some beams which corresponding charge, opposite charge. And after they put those charges on top of those cells, depending upon the fluorescence, they will deflect to the opposite charge and collect it to the beam. And these we are talking about in millivolt range. One, suppose a surface charge is minus 50 millivolt, another is minus 55, another is minus 60, another is minus 65. These millivolt range charge that is put on the cells, depending upon the fluorescent cells, can be sorted out in the different beams. So this is the overview of that. Again, we are not going to ask you about the engineering of that part, but what it will help is understanding the next question, that what exactly we are doing. This is the important question, that we are going to ask a question, that how you are reading the FACS data. Now, this slide, we will connect it with this color, this red and green. So this is just the color wise, this is just an outlier because this is nothing to do with our red green, but this slide, we will connect it with this slide. So we have a green fluorescence and a red fluorescence, and see that how we basically plot the data and how it means, and you will have to read some of the data. First, when we have these FACS data plotted, we put them in these 4 quadrants, and there is a naming of this quadrant, and it's by just convention. We call this as this one as a quadrant 1, quadrant 2, and then 3, 4. It just doesn't go clockwise. It doesn't go anti-clockwise or anything like that. It is just goes the row wise. So the top one is Q1, Q2. The bottom is Q3, Q4. All right. So what we are seeing, that the origin right here, if you actually see here from here, the red fluorescence will be the minimal. As we are moving through the x-axis, the intensity of the red fluorescence is increasing. On the other hand, here also the green fluorescence is minimal. If we are moving up through the y-axis, your green fluorescence is increasing. So what is happening, that when we are collecting the cells, see that the fluorescent light detector is collecting the cells, which does now think about that. This is replaced by your green and the red fluorescent, those kind of proteins, and the non-fluorescent protein. So here it will be replaced by those green and the reds. And they are detecting what's passing through. And the scattered light detector is also detecting what is passing through here as well. So what will happen, that when you pass these cells and plot their data, if you have the cells which doesn't have any fluorescence, that means that here those are the blue cells, here these are the blue cells, there will be nothing detected on the fluorescent light detector. Remember that. Nothing will be detected on the fluorescent light detector. Sometimes there could be passively some antibody is riding through, is just attached. You may have a little detector, but your scattered light detector will detect that there is a dark spot in there. That means a cell without any fluorescence passing through. So they will all accumulate together, and these cells will be right here. So ideally, all the cells should be right here at 0 and 0. That means that these cells doesn't have any fluorescence whatsoever, ideally. But experimental wise, that you have these antibodies in the solution, and one antibody doesn't have any affinity or marker, but it just can stick to it, and it can just have a slight fluorescence or background. So experiments doesn't work perfectly. So that's why these non-fluorescent cells are in here, but you can see that they will have minimal red fluorescence, minimal green fluorescence. So you can see that right here, there are some outliers. And sometimes what can happen, just by chance, a couple of more antibodies got trapped onto the surface of those cells, and it just gave a little bit of more green fluorescence here, green fluorescence tap, green fluorescence right here, red fluorescence trapped on top of those, and it can just be outliers, some of them. But what happens, that if you have a T cell, remember that it should have the red and the green, both of them should be on them. And now what you see, that these cells right here, so that means that if I just go here, the red fluorescence increasing, so more red fluorescence, green fluorescence increasing, more green cells. So these are the cells which have greater red fluorescence as well as green fluorescence. And that's why we are finding the cells in here. So we will collect these cells from those beans, because these will be collected in the specific bean with the red-green fluorescence. That's how you program the cells. These will be all collected from this drops with no charge right here. And these will be collected. And this will be your pure T cells. And what we will tell you, that we can give you some hypothetical questions like this. sometimes you may find, so you can find some of these cells right here. So technically, if you find some cells here, there is high red fluorescence, but no green right here. If you find some of the cells right here, these will be high green fluorescence, but no red in here. So these things, so how do you know where to draw the lines for the quadrants or the exact location for the lines? So this is basically a software that is supplied by the instrument, and the instrument calibrate that depending upon the charge that they are putting onto the cells, and depending upon the charge, they can manipulate these lines, that whether this will go in this direction and this good direction. So this is depending upon the charge they are putting it in. So very sophisticated software. These are proprietary software. See, if it's a Beckman, they will do this in a certain way. If this is like a Carl Zeiss, they will do it in a separate way. So depending upon what instrument it is, what it is, but these are their proprietary software, and they will do it. You can manipulate them using the software interface, that how separate you want, whether you want this one like this and this one like this. They are like really widely separated so that there is no contamination in between, depending upon that. We only want the cells with the both fluorescence in high amounts. So these are the cells with high red as well as high green. See that these are some high reds, no green. I will not take these cells. These are some of the cells right here. Will not take those cells. If you have a hypothetical cells that only expressing red fluorescence, they will be in the quadrant 4. Hypothetically, if it's just only expressing the green fluorescence, that could be in the quadrant 1. Those kind of question, it can happen. So sometimes it can happen that sometimes if you're using a monoclonal antibody that is bad, that is not reacting, you have these two sets of antibodies. And two sets of antibody. One is a CD3, one is an antibody 1.2, and usually these are monoclonal antibodies. And suppose you're running effects with those two, and suppose all your cells are just instead of here, they just all your cells that you are seeing, that all your T cells ended up in here. So you want to better check the green antibody, which is right here. You want to check the green antibody, whether your anti-Thi 1.2 antibody is perfect or not, whether it's working or not. You may want to change those. So those are the things that you can troubleshoot, that can happen over the period of time, that you are expecting this result, but it just showing you some weird thing, that where it should be like here, it's not here, but here you can do this. So this is one of the type of question that we can ask, is that suppose your T cells supposed to be here with this red and green, exact same experiment, but unfortunately you see that they are suddenly in this area. So in that sense, you check the green fluorescence and the anti-Thi 1.2 antibody. On the other hand, if you just end up seeing that you have doubly labeled that during the experiment, and they're all ending up right here in the Q1 quadrant, and at that time you want to check that what they are right here in this condition. This could be one set of the problem. So we can see that your one set could be number 1. We will say antibody problem. This could be a problem. Now, there could be another interesting thing. What happened? That expression means that these are the proteins that are present as an anti-CD3 or anti-Thi 1.2. Suppose you have a mutation in the T cell, and it is not producing Thi 1.2 anymore. Expression level of Thi 1.2 is not there, so you will find those cells right here. On the other hand, if we just go back in here, if you have an expression of this CD3, this suffered from some kind of mutation, there is no CD3 expression, you will end up getting the cells from here, because this is only the thing that is working. So these are some of the type of things that you can think that it could be an antibody problem. It could be also the T cell expression mutation problem, if those kind of things are given as your distractor, that suppose you find some of the cells here, some of the cells here, suppose you find cells right here. And if we just say that maybe the cells doesn't have any Thi 1.2 expressed, that could be one of your answers. or it could be your antibody of the Thi 1.2 that is not working properly. That could be your problem as well. So if both of them are there, both could be the answer. So both will not be there. Or if both of them are there as an A and a B, then we'll say A and B both could be the probable answers. So these are some of the kind of a mindset or thought that you can just think about extrapolating to a lot of different these questions, that how you basically analyze and rationalize those questions. That will be helpful. That's why we spend a good amount of time on these. You will see that we will spend a good amount of time in some of the specific slides, and then we will move comparatively faster through some of the slides where you will not have that many critical thinking questions and such. Say, for an example, here we are looking at a very straightforward question: growing cells in 2D, 3D culture can mimic the tissues. So what we mean by these, that these cells that are grown in here, these are the epithelial cells. So the epithelial cells, when they are in the tissue, they do have two different environment on their apical and basal surface. What we mean by these is think about that if you just take your intestine, this is suppose this is the intestine, and I'm just saying that just to making easier, this is the hollow, or this is the lumen of the intestine. This is the hollow part of that. And here you have the epithelial cells, and one of these cells is right here. Now you can see that this cell is sideways, so I can just draw it like here. So this is the cell that is sitting right here. So this is the apical surface for that cell, the top. This is the basal surface for that cell. See that right here, you have an apical medium and a basal medium. So that cell, the top part where you have these microvilli, this is projected into the lumen where your food material and everything pass. This will be the same thing. Of course, it will not have microvilli if it's a blood vessel, but this is the basal surface. You have an entirely different environment. So if you're trying to culture these cells, it actually, rather than just growing them in a regular media on a Petri plate state, if you have this 2D culture, this help in the polarization of the cell. What is a polar cell or polarized cell? These are usually epithelial cells. See, this is north, and these are the two poles. This is here, this is your apical pole, and this is your basal pole. So these are the cells which does have two directional environment. If you just take a red blood cell or a T cell, see that they are moving through the blood, or the white blood cells, red blood cells, they are moving through the blood. Really, they do not have any specific pole. All of the sides are facing this liquid environment in the same way. They do not have the polarized cells. So, but most of the time, the epithelial tissues will have these polarized, one apical, one basal surface, and a 2D cell culture helps to polarize them in that fashion. You have a different environment to the basal surface. This is a basal media, and you have a different environment in the apical media. The pH could be different. The ion concentration could be different, and those tiny differences could be there. All right, so that's all we need here from here. So this is, again, 3D culture. Also, people now do organoid culture. So this just shows you just a fluorescent label. So this red middle ring is a hollow structure of a lumen of an intestine. And you can see that right here, the green lines and the red lines, there is a tiny green line at the bottom as well. So the basal and the lateral surface, they are fluorescing green, and the apical surface is fluorescing red. What does that mean? And we'll make those sense at the later part, in the part 2 mainly. So the blue things are the nucleus. So you have some specific proteins that are present on the apical surface of these polarized cells, but the lateral and the basal surface, they have a different set of proteins, and they are marked by specific fluorophores conjugated with antibodies, as you can see that. So we are going to see that depending upon the function, you have different transporters. This is mainly the module 2 content. You have a specific type of transporters only restricted to the apical surface, and you have a different type of transporters which are restricted to the basolateral surface of these cells, which makes the cell polarized. So just an overview of that is good enough. All right. So here we are going to spend a good amount of time on these hybridomas. and we'll go one at a time over this. And this is one of the most confusing parts sometimes, and it just takes a little bit of time. So how we make these monoclonal antibodies that we hear all the time. Understand that what the monoclonal antibody is for. So if we just take up proteins, and this could be anything. This could be a viral protein, it's a bacterial protein, your own protein that you need to tag for therapeutic purposes. It could be anything. So this is essentially a protein. And usually a protein will have the amino acids which are buried inside and not exposed. Those usually cannot be seen by the antibodies. Most of these residues of the amino acids, which can be seen by an antibody, are usually on the surface. Now, an antibody can bind to many different places depending upon that. And these are called the epitopes. So epitopes, again, I'm reframing that the epitopes are a stretch of in between, say, 5 or 15 amino acids, which can be recognized by a particular antibody. What we mean by this particular antibody. So here we are seeing that, okay, all right, one antibody recognizes this green, and one can recognize that the blue, one green, one can recognize the red, one can recognize the purple. You have 1, 2, 3, 4. So 4 different epitopes, 4 different kind of antibodies can recognize these. Why? Because all of them have different amino acid sequences. Their folds and structure, or the three-dimensional structure, is not the same. All these 4 epitopes differ in their three-dimensional structure, and you have a corresponding exact same groove on the antibody arms to fit exactly to the corresponding. So you have 1, 2, 3, and 4. So where these comes from, remember that these antibodies, these are secreted by the B cells or the B lymphocytes, not the T cells. The T cells are usually called the cytotoxic, or the different types of T cells. They are regulatory function, directly kill the cells. But the B cells or the B lymphocytes of your immune system, they're a type of white blood cells as well. They secrete the antibodies. So one B cell clone will secrete antibody type 1 just for making billions of these different ones. But this another one will do 2, another will do 3, another will do 4. Same B cell, the same clone of the B cell, this number 1, this will only make this epitope against 1. This will not make 2, 3, 4. The ones which, if we just change the color, the other one that will make 2, not make 1, 3, and 4. So each one of the B cell clone is unique for just only one type of antibody, one specific type. And this is an amazing thing that beyond our scope, that when there are some specific genetic means by which these variety of these antibodies molecules are produced in the B cells to fit basically billions and trillions different types of combinations, and one will fit to one epitope no matter what. So all these different arrays of B cells and the antibodies are coming together and trying different ones, one will fit. Now, your goal is that you want only the monoclonal antibody, that's why you say monoclonal, against the red epitope, as we just going through this one. Only the red epitope. We do not want any antibody against any one of them. So to do that, you have to isolate the B cell, which is producing the anti-red epitope antibody. That is your goal. And that is what we mean by generating a monoclonal antibody, that from the mouse spleen cells, we are going to select only the antibody producing against one epitope that is a monoclonal antibody, and have huge implication in therapeutic purposes, because some of the cancers actually express a particular epitope for a particular mutated protein. You can bind toxin to that or imaging fluorophore to that to image those cancers and do those. Huge amount of application from therapeutics to diagnosis. So huge amount of very price technique. But so that's what we are going to do in the long term. All right. So what we do is, like, you have the mouse spleen cells, the some cells which are red, that is making antibody to the antigen X. that we just made, and that is your X. So we are trying to do from here that first we take the mouse spleen cells. These are all your B cells, and we can just easily separate them out using the FACS because you have this marker protein. You run them through the FACS, and you can collect the B cells. Now you have some mutant mouse myeloma cells. These you get from another lab or an industry. These are commercially available, and they usually come with two different types of mutations or both of those mutations, and I'm going to come with that. So they have some kind of mutations, but these can grow in a selected media. So what you do that you mix the B cells with these mutant myeloma cell lines. And again, myeloma means that this is a kind of a leukemia, a cancer from the bone marrow, which essentially, but these are the cancerous myeloma cells of the B cells, but they do have the mutation, cancerous mutations to make them immortal. And that's the idea. So now you take the two cells and use the polyethylene glycol or Sendai virus. These are called the fusing agents. So they fuse the cells and their nuclei will fuse as well. So they will have their genetic material, they will get combined together. Now what they do that you take a particular media, and it's called a HAT media, hypoxanthine, aminopterin, and thymidine media, and you grow them. What you are going to see that the unfused cells are going to die, which include the non-B cells, and the fused cells are going to grow. So we are going to collect the cells which only make antibodies against the red. The fused cells could have blue, green, and the purple as well, but we will just screen the cells which are specifically making the anti-red antibody, and we can just screen them out and grow them. So that's the bigger picture. So first, let's see that what you have in this media. You have a hypoxanthine. This is a precursor of this is a nucleoside. Then you have an aminopterin. This is an inhibitor of an enzyme. And thymidine, this is also a nucleoside. So let's take a look at that what you have the media and how the selection process essentially goes. So let's take a look at this. So when we call a nucleoside, this is just a little bit of recap. If you just look at the DNA, the ATCGs, so right here, you can have the base A, T, C, or G. Here you have the deoxyribose sugar, and then you have these 3 phosphates right here. So you have a 1, 2, 3 phosphates. If you have this, we call this as a nucleotide. You have the nucleotide. So you have the 3 phosphates. If you do not have the nucleotide, you have just these bases right here. These are the bases right here, ATCG, any one of them, and the sugar. So sugar and the base right here. This is a nucleoside. This is a nucleoside, which is a precursor of the nucleotide. Now remember that this deoxyribose sugar and the base, this can be given to a cell, and this can pass through the plasma membrane into the cell. This can cross the plasma membrane. As soon as you put the phosphate in there, the nucleotide, they will not be able to cross the plasma membrane. These nucleotides must be synthesized inside the cell because these phosphates are polar. They will not go through the plasma membrane. This is completely hydrophobic, and it can carry these deoxyribose sugar along with that through the nucleoside. So nucleosides can cause the plasma membrane. If you add them to the media, the cell can take them up. Nucleotide, they cannot take the cells up. This is some background just to understand the overview of the process. All right. So your nucleosides are just the sugar and the base. Now, to make the nucleotides, to make the DNA, you need to make the nucleotides. And this is your AT, and the C and the Gs, of course. AT and AT, C and the Gs. But here we are only interested in 2 of them, the others, of course. But here we are just only interested in the guanine right here and thymine T. We are messing up only with those. This, if the adenine is made and the cytosine is made, we are not worried about those only thymine, and that's how the experiment is. So let's see that how the nucleotides can be made. So in the cell, there is a de novo pathway. What means that this is the from scratch, de novo, from scratch. The cell, you give them sugars, you give them the amino acids, and from the sugars and the amino acid, the cell is going to make the nucleosides. They are going to go through an enzyme called dihydrofolate reductase, and this is going to be transformed into the nucleotides. All of them, remember that, ATCGs, the cell is going to make that from the scratch, from the sugars and the amino acid, and make the DNA, and the cell can survive. Now, aminopterin is a drug that inhibits the enzyme dihydrofolate reductase. So if you add aminopterin in the media, which the HAT media contains, this de novo pathway will not function. So you kill this pathway. So the only way is the Savage pathway. There is an alternative pathway that can actually work through this. But there is one issue with that. If you just give them just only sugars and the amino acid to the cell will not survive. You have to give them the nucleoside, which is already made and supplied by you. So because you have blocked the pathway of nucleoside synthesis. So where is your nucleoside? Hypoxanthine is the nucleosides for guanine. So to make the guanine or the G, you need the hypoxanthine. Thymidine is the nucleoside to make the T or the thymine. So these are your precursor, thymidine and hypoxanthine. So what you do that you actually put, if you just go back to the previous slide, see that what we have put in the media. We put hypoxanthine. This is one of the nucleoside. We have put thymidine. This is one of the nucleoside, and this aminopterin, which is the inhibitor. Now what you have, those two nucleoside in the media, you have put aminopterin, so this pathway will not work. But the cell, in a normal cell, it will have an enzyme called thymidine kinase. So this nucleosides, you just add it in the media, it will go through the plasma membrane, it just ends up in the cell. And in the cell, you have the thymidine kinase. That's going to put all the 3-phosphates that we talked about. You have the sugar, the base, and the 3-phosphate. Thymidine kinase is going to put the phosphate. Now that nucleoside becomes a nucleotide. So your thymine is delivered, no problem. Thymidine kinase will make the thymine. So your T is delivered. You put hypoxanthine. The cells will also have hypoxanthine guanine phosphoribosyl transferase. That's the enzyme. That's going to put all the 3-phosphates on the guanine. So your G is delivered. And of course, the A into the media as well for those. This is not mentioned, but you have to put those as well. But we are not talking about that. No, A and C, adenine and cytosine. So you have the thymine produced and the guanine produced. It can make the nucleotide and the DNA. So this is all good. So what happens, the take-home message from the first part is that if you take the aminopterin and kill this pathway, the only way a cell will survive, that you have to provide them with the nucleoside if you kill this pathway. That's great. So now let's take 3 cells right here. 3 cells that we have, the 3 scenarios from the previous part. One is a myeloid cells. This is the mutated cells, cancerous cells that we are fusing. This is the B cell. And this is the fused cell. This is the fused cells, which is the myeloid cells plus the B cells. And this is your hybridoma. This is your hybridoma cells. So what are the different things? What is happening? So in the myeloid cells, you can have 3 different kind of myeloma cells, and you can use any one of them, whatever is available to you. You can have a mutation of the TK. That means we write it as a TK minus. That means that the cell cannot make thymidine kinase, this myeloid cell. Another type of myeloid cell, you can take an HGPRT minus. These type of cell will not be able to make the AGPRT. And then you can have a double mutation, which you can have a TK minus plus HGPRT minus. You can have, and your choice. Any of these three types, whatever you use, the outcome will be essentially the same. So if you take these myeloid cells and grow them, you always have to grow these either using, so they do not have this, either you have to build them or grow them, you have to grow them in the de novo pathway so that you just provide them the sugars and the amino acids, and there is no aminopterin there, they can make the nucleotide. That's why these myeloma cells can grow. Now you take an het media, remember that, you take the het media which contains the hypoxanthine, thymidine as a nucleoside and aminopterin. So you kill this pathway. The myeloid cells with the TK minus will not be able to make T. The AGPRT minus will not be able to make G, and this one will not be able to, the double mutation will not be able to make T and G either. So if you grow the myeloid cells in the het media, no matter what, it will die because it cannot make T, it cannot make either T or G or T or G both because you have the aminopterin in here. Now we go to the second one in the B cell. In the B cell, if you're just growing into the het media, great, aminopterin is added, so it will not be able to use pathway. This is blocked, so you cannot use that. But the B cells have TK plus. It has a functioning TK. It also have AGPRT plus. It has also have functioning AGPRT. So this pathway is great, perfect for that because you are providing thymidine, you are hypoxanthine. B cells will survive and make nucleotide. Problem? This is a mortal cell. After those 25, 30 passages, they will die eventually. If they are not fused or with the myeloid cells, they have a mortal lifestyle. Myeloid cells are immortal, but as they do not have the enzyme, they will not be able to survive. Now you have a fused cell, which is the hybridoma. From the immortal myeloid cells, the immortality or the cell cycle, that immortality comes from the myeloid cell. And the B cell provides the complementation of the AGPRT. It now, no matter what it is, it becomes now TK plus and AGPRT minus. If it is this, TK plus is produced by the B cells that will complement it. If it's AGPRT minus, that is produced by the B cells that will produce. If you have a double mutation, both of these genes are normal in the B cell and they will complement that. So the fused hybridoma cell will be able to use this pathway to make the nucleotide and will survive. Myeloid cell will die eventually. B cells are mortal. They will die eventually. So now what we'll do that you have these hybridoma cells and there will be several colonies of the hybridoma cells. You have a colony here. Suppose this is the Petri plate. You have a colony here. You have a colony here. And you check each colony which is making antibodies against the red epitope. And you found that this is making the red epitope. You take this cell out and that is your hybridoma. You will now grow them without aminopterin in the regular media, and it will be absolutely fine using the de novo pathway, just regular sugar, amino acid, no hypoxanthine, nothing has to be added in the regular pathway. This will just keep on producing your red, anti-red epitope and monoclonal antibody forever. You can culture them in 250, 500 liter large fermenters or animal cell culture media, and very expensive for producing these large sets, but you are good to go, and you will be able to produce these in very large amount for the commercial purposes. So that's basically the entire part of that. So I also made these some of these supplementary videos because this is some of the things that where the questions comes, and it could be a little bit tricky. So we can ask different types of questions, but sometimes remember that this is the take-home message from this slide, that what you do, that remember that that if you have a deficient gene mutation like a thymidine kinase or a GPRT in your myeloid cells, that is provided by the B cells. So B cell is complementing with that. So now what happens that suppose we just make another addition to that, like so you have a dihydrofolate reductase, so which is right here is dihydrofolate reductase, DHFR. And I say that your myeloid cells, they also have a mutation on the DHFR. And what's going to happen to those cells? So if you are having a DHFR, remember that you have to constantly, it is very, if you have a mutation like that, you have to provide those cells in a special condition. You have to grow them because it is not easy. You have to provide them with additional nucleosides so that they can bypass this to make those myeloid cells survive. But theoretically, what can happen that if you have a mutation in the DHFR in your myeloid cell, if you have the B cell is going to complement that the same way as TK or a GPRT. So whether it is an X, Y, Z mutation in the myeloid cell, and suppose that is a problem for that is normal growth, you are just using those as a nucleotide supplemented media. You can use those cells in the nucleotide supplemented media. You can use some of these carrier transporters, which will supply the nucleotide to the plasma membrane in lipid nanoparticles to keep them alive. There are many different ways that you can do, but that is not important. The thing is that if you have a myeloid cell with X, Y, Z mutations and you are keeping them alive, and if we just ask them that we are just using them to make these hybridoma cells, what happens? Remember that wherever you have these X, Y, Z mutations, no matter what, doesn't have to be TK or a GPRT, everything can be complemented by the B cells because your B cell has healthy X, Y, Z no matter what. So we can actually ask you some questions that other than a GPRT or TK, we have some other mutations like in an X gene or a DHFR. And what's going to happen? It's going to grow the same way and your selection should be absolutely fine. So say, for example, we have a DHFR mutation and we are somehow growing those myeloid cells, but this DHFR is going to be replaced by the B cell. So after the hybridoma selection takes place, you can still grow those hybridoma cells using sugars and the amino acid because the myeloid cells did not have a DHFR to be grown on those, but that is supplemented just like TK and AGPRT by the B cells. So these are some of the critical thinking questions that we can ask. Remember that it could be A, B, C, any gene, but that just always remember that the B cells will have a healthy set of all the genes required for a normal cell to function. So whatever a myeloid cell is deficient, it's always going to be supplemented or complemented rather by the B cells. So your hybridoma should be fine. So that's basically your take-home message from them. So I think the next slide, just an overview of that. Sometimes we call as in plasmacytoma cells. Here, this example is just with a TK minus. So you can see that TK minus means it doesn't have that. Fuse has a TK plus, TK minus. What it means that you know that you have two alleles of the chromosome. You have the chromosome 1 and 2. And even if there is one is defective, one is plus, that means that that is enough for making the thymidine kinase. So this TK minus is complemented by the TK plus and is good enough. And TK plus is, of course, the mortal splenic cells that will die. These are some of the parts that usually you are going to see the most intense type of critical thinking questions. So I make sure that we spend a good amount of time. All right. So, but out of all those, just remember the PMSF. We are going to find these usage at the very last few slides when we basically make purify the organelles from the cells. So we are going to talk about the PMSF, phenyl methyl sulfonylureide. Remember those. And why I put all these different names in here, because we are going to use all of them in different places all throughout the chapter. We're going to use those. And this PMSF and the monoastrol, these are some of the drugs we just use and that's all. So this is a monoastral. Just remember that what it does. So here you can see that on the spindle fiber, you have the chromosomes. They are just getting separated before this is a metaphase plate. Before anaphase, they are going to go to the poles. But when you just use the monoastral, it just disrupts the motor proteins, which are responsible for moving those chromosomes. And you can see that the chromosomes are all scattered all over the place. And basically that will cause the cells to die because it cannot divide anymore. So these are anti-cancer drugs. Okay. All right. So let's go to the light microscopy. So these are all good. I think that we will stop at the ones which are critical. So magnification, we give you guys very simple magnification. So just multiply these two, objective and the projection lens or the eyepiece power, and you will get that. That's good. Lambda. So usually this is our visual range, about 0.45 approximately, 0.45 to 0.7. That's a visible light range. And when we'll calculate the resolution, you will remember that we usually give you the value, but usually it will be the lowest visible light in this range as well. So these are all pretty self-explanatory. So remember, and just the definition that just what is the best definition, and the correct answer is the distance between the two distinguishable objects, just exactly how it is. That's your resolution. Very straightforward, nothing. And then, of course, you will have these in front of you as well. So definitely remember that how you calculate the D, or the resolution, is 0.61 lambda in sine alpha. And we are going to give, you do not even have to calculate the sine alpha. We are going to give you the value. And we'll give you that exact value. And just problem that we had just given right here, we are going to give you the shortest wavelength, lambda 0.45, immersion oil, the N, and alpha, maximum alpha 70 degree, and then sine 70 degree, which is the N sine alpha. So you have the N, the sine alpha, which is sine 70. We're going to give you the value. So you do not have to calculate even the sine 70. So you just plug into this D equal to 0.61 lambda over N sine alpha. 0.61 is your constant, and then 0.45 is the shortest wavelength. 1.5 is your N, and your sine alpha is sine 70, which is the 0.94. And here you come with 0.194 micrometer. So if you just look at this, this will be approximately, you can see that point, we just say that optical microscope has approximately 0.2 micrometer. That is the resolution. And that is why that's what is ever. So just remember that when we can give you different types of oils where this N, the best media, this will change. You will see that this will change. This could be 1.2. This could be 1.6. Or I'm saying that 1.2 or lower, 1.6 or higher than 1.5, lower than 1.5. Depending upon that, this will change. So remember that theoretically, if it's 0.194, if you just go to say 0.3, then right here, you have your resolution is actually worse. Remember that, because you are not able to distinguish that good. So your resolution is worse. So if there is an immersion oil that makes your resolution 0.3, that's a bad immersion oil. On the other hand, theoretically, it's difficult to go below that. But theoretically, if there is an immersion oil that you have discovered that pushes the resolution to 0.150, so see that right here is 0.194 originally, and it pushes to 0.150, which is lower than that. So this will be by a better immersion oil. Probably not possible to do that, but practically, but theoretically, just remember that any number which will go over this or larger than this, that will be a worse resolution. If you get a number lower than this, it will be a better resolution. The lower the number, that is a better resolution. That means that you can distinguish in between that tiny difference. So remember that the difference with the number is inversely proportional. Bigger the number, worse the resolution. Lower the number, better the resolution. That's your take home. That should do it pretty much. Straightforward. This will be in front of you. You can calculate that. Simple arithmetic. You will see that. All right. So after that, we are going into the bright field microscopy. There are lots of variations. And the advantage is that simple, inexpensive, no staining required. Now, Here, a lot of people get confused with this, that we say there's no staining required. Now, when we use a DIC or the phase contrast, usually we do not stain them. But what happens that we use this no staining only for live cell imaging in cell culture, where you have 1-2 cell layer thick. You can only see those with no staining. On the other hand, see that you have we are saying that it's a very low contrast for mold biological sample, low apparent optical resolution due to the blur of out of focus material. So how we bypass this? So see that right here. If you are doing the cell culture, you have to check your cells in the flask or the dish every day. And for those, we do not stain the cells. We use the bright field microscopy and we can see the cells. If you're just seeing the cells with the bright field microscopy, see right here, we are looking at those and very low contrast resolution. You really do not see that well. So we use usually a phase contrast to see them. You can see them brighter, DIC, without the staining. This is your optical enhancement. So on the other hand, suppose you are given a block of tissue. This is if you're a cytopathologist or a clinical pathologist, and this is your biopsy sample. In the biopsy, and this is basically if you destroy the tissue, there is no problem with that. But if you just take this piece of tissue and put it under the microscope, you are just going to all see black. You have multiple layer. It's a thick tissue. No light is going to pass through it, and you are not going to see anything. So what you have to do, you have to section it. You have to cut it in a thin piece so that the light can pass through it. All right, you sectioned it into a single layer so light can pass through it. This is your physical enhancement, sectioning. So you section, okay? Even after section, there is a problem because what is happening, it is still a low contrast, most biological sample. A single cell, light will pass through it, but you will almost see nothing inside the cells. So just a physical enhancement of your sample is not good enough. You still have to do a biochemical enhancement of that. This is your staining. So these are the modifications that you do. Optical enhancement is your DIC or phase contrast without staining. Physical enhancement is basically a bigger tissue to section that so that it is light can pass through it so that you can visualize that. And biochemical enhancement will be your staining process. And this is basically what you do. So the physical and the biochemical enhancement we use with the dead samples or the samples in the process will be dead. On the other hand, if we are just doing the optical, remember that we just use these in the live cells because when we are doing the cell culture, we need to check our cells and we do not want to section or do biochemical staining of the cells every time that in a daily routine that we have to do it in the cell culture. We do not do that. So we just use a better method to see these. And all these optical enhancement could be in the same microscope. Remember that they are not separate microscope. You just only need, so you have a bright field and the phase contrast can be built in the same microscope. You just have to remove some of the rings in here and there. So a typical question that we ask, suppose you have a bright field microscope, and if you need to make that as a phase contrast microscope, which are the following things you have to purchase or the additional parts that you need? You need a faceplate objective and an annular diaphragm. These red things are the things that you need to convert a bright field to the phase contrast. These are just the accessories that bright field phase contrast capable microscope that you can have. So you can just purchase them without the accessories, but it will have the ability to add those. This is just, think about that you have a interchangeable lens camera with, you can put filters in there and these and the different types of lens like a DSLR camera or that kind of camera. These microscopes are with the capabilities of phase contrast as well. The same way, the DIC or the Nomarsky, the same way you will see that I have put some supplementary video to see those. You can just have these Nomarsky prisms in there and you have two sets of these things and you can just move those in there and your bright field microscope can work as a phase contrast microscope or it can work as a Nomarsky microscope. It is the same set of microscope. You do not have to have a separate bright field, separate phase or separate DIC. Yeah, if it is a fluorescence microscope, usually it's a separate microscope. But for all the different light microscopes with these are just attachment and enhancement. Or of course, it has to have the capability of doing that. If you have just like a low-cost lab microscope, which may not have the ability to add those accessories. But if you have a higher-end bright field microscope, you have the option to attach all of them in the same microscope that we use. So on a day-to-day cell culture, you cannot see the cells that well, but in phase contrast or DIC, you see them a little bit better. So these are some of the understanding. All right. So after that, these are your physical and biochemical methods for images. So fixation, embed and section, stain. So you can see that you can fix the sample so that when you section them, they don't fall apart. You can use glutaraldehyde, formaldehyde, partially impermeabilize the cells for staining. And then you put them in using wax or resins, and you will use different things for your electron microscopy. And then you take a microtome, you just cut those sections with these diamond knives. Then you have these microscope slide, you put those sections in there. They will stick in there, but now still they are not done yet. This is the only part that is the physical part. Now you have to stain them. So these, you can see that this is a typical hematoxylin and eosin. We call it H&E. This is a gastric mucosal parietal cells. Now, note that your textbook, not only the 8th edition, I'm surprised that they did not change that in the 9th edition, has a print error saying the opposite. It says the hematoxylin is acidic and negatively charged, and it says the eosin is basic and positively charged, which is wrong. So this is correct information. So your textbook has a wrong line in there. So it's not been corrected from that edition carry forward. So just make sure that this is correct. So what kind of question that we can ask? We can ask a question. Suppose you have a patient from where you get a biopsy sample. And if we just, so suppose a small tumor is excised from a patient's body. And then a cytopathologist wants to examine the size, shape, and the arrangement. That's exactly, you are seeing the size, you are seeing the shape and the arrangement of the cells within the tumor by routine histology, which is the study of the tissues. By fixing right here, if you just go right here, if you just see that right here, by fixing and staining this. And for this purpose, what kind of microscopy that you will use? You will just use a regular bright field microscopy. So the bright field microscopy will have these bright background, and it is just the regular bright field microscopy. Just this. This is unstained, but in a bright field microscopy, this is perfectly stained. You will use this method for this purpose. So this is another example, bright field microscopy. You have a bright background. Remember that. And this is also another staining. Detail of these stains are not required, just an overview of that. This is another bright field microscopy. All of them are bright field microscopy. So you can see that in the stained sample in bright field microscopy, you are all going to see these beautiful colored images. But when you see a phase contrast or DIC, you see a black and white. The question is that if you take a stained sample, can you do a phase contrast? Yes, you can do a phase contrast. It will show, but it will not show really some extra information over the bright field. If you just have a stained sample, right, like this, your bright field is going to give you all the information that you need. If you do a phase contrast or a DIC, Nomarsky, on top of that, it's not going to give you any extra information. They are only going to give you extra information over the bright field if they are unstained samples. Just remember that. All right, so that will do it. And then we are looking at the fluorescence microscopy. So make sure that here the fluorophores are excited at a lower wavelength and they emit a longer wavelength. So your emission spectra is always higher than their excitation spectra. And we always give you a question, and I'm just literally telling this question. I just tell this question to everyone. Sometimes miss those questions. Basically, when I just ask the definition of this. So a fluorescent molecule, fluorophore chrome, absorbs at one wavelength excitation and emits light at a specific longer wavelength. And I'm just literally writing this down. And this, I just do that, how detail you can see, and this is in front of you. I just change this one to a shorter. I just change this one as a shorter as a definition. And a lot of people actually choose that. Remember that this is incorrect. You emit a specific longer wavelength. So make sure that these seems like to be absolutely a mean question to ask. But that's the reason I'm telling you and writing you in there that this question will be there. So don't miss that one. I see 50% miss this question. These are some small things, but you know what? This is why you need to look at your notes and put it on your slide and just make sure that this has been discussed. And Dr. Q told that you're going to be a mean question, but told the answer. So don't miss that. All right. So here also, we go to the next question, and the same type of question that we ask here in the fluorescence microscopy that we ask that suppose you have bright field microscopy and we want to convert theoretically to a fluorescence microscopy, and what are the things that you would like to buy or add to that? And here the same way, the 2 pointers, you need an excitation filter. Of course, you will need a light source, which is usually nowadays a laser source. Of course, you will need that. But if that option is not there, you need an excitation filter and a dichroic mirror. And here, just know the function for the dichroic mirror, what it does. The dichroic mirror usually will allow one wavelength. Usually it is a higher wavelength that you do not need to remember. Just remember that it will allow one wavelength to pass through it, but the other wavelengths will not pass through it, and it will just reflect those. So usually for the dichroic mirror, see that right here, the excitation wavelength is in green. That's a shorter wavelength. It will reflect the shorter wavelength onto the specimen. The specimen does have a fluorophore. It will fluoresce the red, and the red is a higher wavelength, and the dichroic mirror will allow the higher wavelength to pass through the projection lens to the image plane. So that's your dichroic mirror. So it is reflect usually shorter wavelengths or a specific wavelength, but it will allow the other wavelengths to pass through it. So these dichroic mirror and the excitation filters are some of the part that we use in this process. All right. So let's see. All right. So another question that we ask, let's take a look at those. I did not particularly put a star mark in there, but I just in several places as well. So here you can see that there is a green mitochondria and a red lysosome in there. And you get these kind of pictures all the time. And the question is that exactly what is happening. So you have a green fluorophore attached to your mitochondria or an antibody or antibody to the lysosome, and you are doing this. Now, remember that right here, this is the most important part, that each time that when you are doing a fluorescence microscopy, you have an excitation lambda, a wavelength, and then you have this dichroic mirror and this filter that will block all the light except the specific emission lambda, which is green in here. So if it fluoresces red or yellow or even or brownish or anything like that, that light will not pass. You will only see green in the background. Now, here you are seeing red and green both. So what is happening in here? So what happens that first you take the picture with this setup where you see the green, then you change the excitation lambda or the excitation wavelength, and you change the filter so that you would allow the red to pass through, and you will see now reds in there, but you will not see any green, and you merge those images. This is one of the most important concepts in the fluorescence microscopy. You cannot see both the pictures at the same time. You will see the green, you will see the red as individual image, and you will merge on top of each other. And this merging is just by your computer software, imaging software will do that instantly. But each time by this method, you can just only have that because you have a specific excitation filter or wavelength and an emission filter, and it will not allow to visualize two at the same time. Yep. So that's just remember that. These are some of the iron-sensitive fluorescent dyes. These are very straightforward. Then we are going into this immunofluorescence microscopy, where we are. This is probably the next longer. I posted some of these discussions, so make sure that you look at those discussions. Those are very important for your exams, and it will just clear up many different things. So exactly what is the concept of these primary antibodies and the secondary antibodies and the secondary antibodies? Why do we need them? And some of these questions are asked, and I will address those at the end as well. When we're looking at this antibody molecule, these are the Y-shaped molecule, and we call this as this fragment AB. And these two arms, these red areas, which actually bind to the epitopes. That's basically the recognition site, which can take all different shapes and sizes. Then you have this FC, and FC is called the crystallizable part of that, a fragment FC. Now, this part is constant among all antibody molecules for the same species. That means if you take a rabbit and you take out different types of antibodies. Now, remember that, as I said, the rabbit is seeing every day hundreds of different antigens and hundreds of different B cell clones. All are going to make those X, Y, Z, anti-X, anti-Z, anti-PQRS antibodies, all of them from different clones. So their FAB region, this top region will be different, recognizing each different epitope, but each of those antibodies will have a constant FC region. And again, if you're just going into a little bit of detail, not needed in here, there are different classes of antibody, and usually the most abundant antibody is known as the immunoglobulin G, and we are mainly talking about that. And all these immunoglobulin G that is secreted by these B cell, for the rabbit, this FC portion is constant for all the rabbit antibodies, no matter what, across the species of the rabbit. Now, if you change it to the human, among all of us, this FC portion is going to be the same for all the human beings. In between the B cells, in between the human individual as well. So this is the part that is going to change, depending upon what it is. And we actually take the advantage of this to do this primary and the secondary antibodies. And what is the advantage of that? That we're going to see. So now what you have, that we do have a cell where you have is protein X, and this is our antigen. Now we are switching gears. The protein X that we actually had earlier has a red epitope, and we actually raised a monoclonal antibody against that. Here, we are not going to raise a monoclonal antibody. We are going to raise what is called a polyclonal antibody. And why it is so? Because suppose this is your protein X with all the different types of epitopes. We want the different antibody clones. So I'm just drawing it with a different color, say blue right here. And all these different ones bind to different portion of the antibody. And then if they are attached with the secondary antibody and the fluorophore, you will have a better fluorescent signal. So we want many type of antibodies to bind in that, and we want these polyclonal antibodies. So to do that, all you have to do is that you purify these protein X. Suppose this is from an animal other than the rabbit, because remember that this is the basic concept. The protein has to be foreign to the animal that is raising the antibodies. All the mammals will raise these antibodies. Mouse, even chicken can raise the antibodies as well. Even the birds also do have the capabilities of making the antibodies, but it has to be foreign to the animals. So if it is a human protein, we can raise it in a rabbit. We can raise it in a goat. We can raise these alpacas or llamas are used, even horses, cows, any animal that you can use. These are the usual thing: goats, rabbits, even mouse, rats, laboratory rats. You can inject it to them, and they will raise the antibody. You can purify them. So essentially, let's take a look at that we just name this antibody like this. We name this antibody, we say that this is your human protein X. All right. So when we inject the human protein X to a rabbit, it will raise an antibody, and we will tell this antibody, as we will name it like this, it will be anti-human protein X and rabbit antibody. This is the legal full name of that what we have. This is the full. Sometimes we use the short, and that's where the problem is. I'm just going to write. So anti-human protein X rabbit antibody. So what it says, the first part says that what is the antigen, and the second part says that where it is raised. If it is raised in a goat, we'll say anti-human protein X goat antibody. So we know that where the antibody is raised. Anti-human protein X mouse antibody, depending upon where it is raised. And this is your primary antibody. Takes about 8 to 10 weeks. They will bind to your protein. Now, what happens that you can actually conjugate a fluorophore to this, and you can do the fluorescent experiment. But what happens that your primary antibody will be destroyed just by this one application. Whatever you used in your experiment, you have to throw out that antibody. And this is your specific protein. So raising this is expensive, because this is custom made in your laboratory or a laboratory, because your protein could be 1,000 different protein. You are sending this to a laboratory to make it for you, or you're doing in the laboratory. You have to maintain these animals very specific for your protein. So these primary antibodies are expensive. So just to reduce the cost of it, the experiments are done with the secondary antibody, where you can preserve the primary antibody more just to reduce the cost. There is no other reason. You can always add a fluorophore to your primary antibody if you have millions of dollars. I mean, like, not really, but just like saying that if you have that kind of money, or your lab does have that kind of money, you can always conjugate a fluorophore, and without doing any secondary antibody or anything, you can just use the primary antibodies, and that's perfectly fine. There's absolutely no problem with that. So why we do the secondary antibody? Just to reduce the cost, and how we do this. So the first, we already got against the protein X, the primary antibody, and these are the protein X. So what we do that we actually fix and permeabilize the cells after sectioning, and we add this primary antibody. It's shown as this 1 degree. That's your primary. And just we add to the cell, and you can see that they are binding to this. Now we have a secondary antibody that we add to, and this is bound to this Fc portion of the primary antibody. And this also increases the signal of that signal, and this you can visualize. So how we basically make the secondary antibody. So first, see that right here, we have made a rabbit antibody. So as I said, that all the rabbit antibodies, no matter what, this Fc portion of all the rabbit antibodies are going to be exactly the same. So what the industry does, the industry will take a rabbit and not inject it with any protein X, just a regular rabbit, and it will bleed it humanly and take all these immunoglobulins or these antibodies or these IgGs, which are the immunoglobulin, all the antibodies it will collect. So all these different antibodies, all of them will have the same Fc portion, but these AB portions, binding portions, this will be different. And they're going to take all these mixture of these antibodies, they are going to inject it to another animal. So they're taking it from the rabbit and another animal. This could be animal, this could be a goat, this could be a chicken, this could be a mouse. Theoretically, could be a human as well if you want to use a human as a subject, but technically that's unethical. That's why but technically it will work. Goat, chicken, mouse. So other than the rabbit, they have just put that. So essentially what's going to happen, that animal will raise all different kinds of antibody against this part, but that is not what we are interested in. It's going to raise a large amount of antibody against this Fc portion, which is the same in all the antibodies that we have purchased. So now we purify these antibodies from these animals, goat, chicken, or mouse, which is against this FC of the rabbit, FC portion, which will recognize the FC of the rabbit. So that is what we are getting as a secondary antibody. So let's go to the next slide. So when we are going into the first antibodies, you can easily see that it is recognizing the FC portion of the primary antibody. Remember that this is your anti-human protein X rabbit antibody. And your secondary antibody is anti-rabbit antibody. This is the full legal name again, antibody goat. If it is goat, goat antibody. So you have raised it against the rabbit antibody. That's why it is anti-rabbit antibody. And you have raised it in the goat, so it's a goat antibody. So to say anti-rabbit antibody, goat antibody, it's a mouthful. We usually do not say this antibody. We skip these and we just say anti-rabbit goat antibody. If we do not mention the name of any protein or anything, you will assume that anti-rabbit goat antibody means that it is against the antibody of the first animal. So that is the convention. And this is what I put down in the discussion, that we say that anti-rabbit. Sometimes we just even say this, we say anti-rabbit antibody. So what will happen that here, your primary antibody is rabbit. So your secondary antibody, if you have a secondary antibody with any fluorophore with anti-rabbit, which could be anti-rabbit goat antibody, anti-rabbit chicken antibody, anti-rabbit mouse antibody, you can use any one of them with all different fluorophores in here. So this is the overall concept of the secondary antibody. So the question is that why this is useful or where the advantage of that. So think about this way, that a lab works with proteins X, Y, Z, A, B, C, P, Q, R. They are working on these different 9 proteins. So they have to raise, to study them inside the cell or localization, they have to raise primary antibodies against all 9 of them individually. But when, say, suppose all separate graduate students or postdocs or other people are working them in a separate project, separate understanding. But all they can have is that for those, if they're all raised in rabbit, they can just use an anti-rabbit, doesn't matter, anti-rabbit antibody, doesn't matter where it is raised, what kind of fluorophore, it could be green, it could be red or yellow. They can use the same reagent because they are doing separate experiment in separate kind of context. So it doesn't matter if the fluor is the same, but they can use the same secondary antibody for the entire spectrum of their primary antibodies. Whereas each time they use this primary antibody conjugated with a fluorophore, they're going to destroy that in that one step. But if you keep the primary antibody, what you do that you just mix it with the cell, and you can take this antibody out and keep it in the refrigerated condition, and you can use it at least 2-3 times. So you use a very little amount of the primary antibody, whereas the large amount of secondary antibodies are raised commercially in a large amount. So they are much less expensive and reduced. So this is the concept of your primary and the secondary antibodies. The next part. Let's see. All right. So after that, you can see that there could be a primary antibody. We are increasing the fluorescence by binding them to multiple of these secondary antibodies. Also increase the signal as well. And there are could be molecules which can be directly labeled as well. So here are a couple of other nicer pictures. Remember that all these multicolored pictures are taken separately. So this gives you the idea. So the green on the top and the red, they are taken separately and then merge. And these will be the same way as well. All right. So you can also what you can do, that we are talking about these antibodies, and this is a destructive method. You have to fix or permeabilize the section. Now, if you want the localization of some of the cells, which you want to see it in the alive in the cells, what you are going to do. So there are several methods. So here, what you can do that you can add an epitope to a protein. And here, as soon as you use any kind of a antibody, you are going to fix the cell. So it's not going to work. But what you can do that you can fuse your gene with this fluorescent gene, which can be available originally from these jellyfish, and they have been modified by molecular biology technique into all these different fluorescent protein colors. And you have all these gene sequences, and you can fuse them with a gene. And many times, these genes are functional even with these fused genes. So you have a gene chimera, and you can make a recombinant which will essentially express these GFP and the proteins together, and you can find the localization in the cell. So this is a dictyostelium, this is amoeba, and this RAB8 is molecule which is basically vesicle formation and recognition related protein. We're going to see that later. And this is used for the formation of these pinocytic vesicle formation in there. This is black and white, but here the pinocytic vesicle is formed where this dark area is where the vesicle is being formed. Right here, you can see that right here, the vesicle is formed, and here the vesicle is actually engulfed in this process as well. It's still there, but you can see that right here, it's still there, but it just shows that formation is process where this RAB8 is mostly expressed. And after that, it is not required, although the vessel is still there, or the content of the vesicle is still there. So these type of things you can study. And then we are looking at some of the super resolution microscopy. We are looking at some of these deconvolution microscopy. This is a totally digital tool where you can just, by a computer algorithm, you can reduce the background of these fluorescence and then get a sharper image. And again, remember that when you acquiring the image the same way, these are all fluorescence microscopy, all these different color images are acquired separately, then merged, and then you just take out all the background noises from the different planes and reconstruct into a three-dimensional image. All right, so after that, we are going into the LCSM or the laser LSSEM or the laser scanning confocal microscope, which can observe the image in a single optical plane. This optical plane, just remember that this is a slice of an entire image. So if you have a thicker object, in light microscopy, we had to cut it, section it, and then observe that. You can have a thicker object in here, but you can slice it optically by this method. In the confocal microscopy with the confocal pinhole, you get the image from a particular plane. So here, what it shows that this is a plane of focus, which is the optical plane. And this blue rays are just going through this pinhole into the detector, and you are just seeing that. See, the green thing is coming from the out of focus plane, which is a different optical plane, and that is blocked by the pinhole. And this is done by the laser scanning LCSM. Again, direct is not going to go, just this overview of this is good enough. So here you can see a regular fluorescence microscopy. This is the image volume, thick optical plane. Here you have a slice, thin optical plane. You can see our chin egg. You can see that how difference is that in a confocal microscopy. All right, so here there are two different things, and don't get intimidated with the pictures. We are not going to ask any of these components or anything like that. Just a couple of different things that what happens, that this is called a point scanning, where you actually focus the laser beam to a point, and then you go point by point illumination and occurring image in this raster scanning fashion. And this is very accurate, but it takes a lot of time. So the image must be static. That means this is a fixed cell with a fluorophore. And this is called a point scanning confocal microscope. So they scan each of these planes. They can go in the same plane and then multiple plane, and they will reconstruct the image. Takes a lot of time. So the image must be fixed or static. Whereas if you actually use some of these fluorescent proteins, like this that we talked about, fluorescent proteins, and you just want to see that how they are growing in the cell in a dynamic nature, you use this spinning disk confocal microscopy, where you have a pinhole array, and it could be 20,000 micro lengths, which will illuminate all these different spots simultaneously, which will not give you any background. And you acquire this image from different points at the same time, and then computationally basically reconstruct the image. These are for live dynamic specimen. This is your spinning disk confocal microscopy. and where it is useful. See right here, you have some spicules of actin which are fused with the fluorescence protein is growing. So if you just look at that right here, there is a spicule right here. Right here, there is one. This is a little bit longer. This is a little bit longer, even longer, even longer. Or see right here, there is almost nothing. There is a tiny spicule growing here. It is longer, 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 longer. So you can see that there are some actin filaments, microfilaments growing inside the cell. And it's within 140 seconds, you can image that. You cannot capture this with a point scanning because this is much, much slow. You have to use a spinning disc confocal microscopy. So the only take-home message, where you use a point scanning, you use it for static specimens. Where you use a spinning disc, you use for a live dynamic specimens. That's all you need. 2-photon excitation microscopy, this actually fires 2 photons and they reach within a femtosecond time. So that's 10 to the power minus 15 second. So here is what is interesting, that we always know that your excitation spectra is lower in the nanometer. So it's a shorter nanometer, shorter wavelength, and your emission spectra is higher. Here you can see that 960, this is double the wavelength, and this is in infrared. This is not in your visible wavelength, but here the excitation spectra is longer than the emission spectra. So you may think that, okay, all right, this is not following the fluorescence microscopy. But again, what happens that when the 2 photons reach the fluorophore within the femtosecond time, they think that these are coming at the same time and the energy is additive. So 960 plus 960 reduced to half, which is the same energy that the 488 nanometer gives. It is the same kind of energy to deflect the electrons. And when the electrons lose their shells, they're going from the, again, I'm not going into the chemistry or the physics of that. And when they are just coming down the shell, they are just releasing the energy, which you see as an emission. But again, this is also, I explained that this is the 2-photon microscopy. They reach almost at the same time. So this additive nature brings them down to the same energy to do that. What is the advantage? If you use a blue light, see that when it is moving through the media, this is a visible light, it just diffracts so much. You have so much of background. But see that what happens in 960 nanometer. This is infrared. This is invisible to your eye. So there is absolutely no background as it is moving through the media. So, but it excites the fluorophore. You get only the fluorescent excitation. So if it is a thick specimen, you can point it at a focal plane without getting out of a focal plane fluorescence at all. So you can use this for a thicker specimen up to 1 millimeter. So you can see that you can just do this mouse brain live specimen visualization. These are some of the neurons of a lobster. So you can just take a look at this your textbook for these images. So these are the advantages of these. So this is TIFR and this is epifluorescence versus TIFR. So sometimes people just ask, is that what is the epifluorescence? All the fluorescence microscope where you have these objectives right here. Let's go back all the way to the fluorescence microscope picture right here. See that right here, you have the specimen and the lights come through the dichroic mirror projection lens and the image plane. You are seeing this from the above. This is your epifluorescence. Epi means above. This is your fluorescence, and all the fluorescence microscopy, including the confocal and everything that we discussed, they are all the same. Only right here that we are seeing in the TURF or the TIRF right here, where you are seeing the epifluorescence in a little bit different way. So it is the light is coming in an angle and it only illuminates the total internal reflection. It only eliminates just only a tiny part of your sample. So you can see that right here, your sample has this much of thickness. And this top portion, which is known as evanescent wave, this is just only 50 to 100 nanometer deep. Your sample may be 2,000 micron, 1,000 nanometer. It could be 1 micron, 1,000 nanometer, but it's only illuminating just only the portion of that, which is called the evanescent field, so that only the specimen closest to the cover slip is illuminated and not the background. Because if you can see that right here, you have an epifluorescence right here. And this is how some of these microtubules look like. When you do a total internal reflection, you can see that these are illuminated, these are not. What it means here, it looks like that everything is on the same plane. Whereas here, it is telling you that this left part is closer, which is closer to the cover slip. This part is actually deeper. This information is not available from an epifluorescence, which you can get that where the depth perception, you can get that whether it's closer or deeper into the tissue by these total internal reflection microscopy. All right, let's moving to the fluorescence recovery. So here in this process, we are using some of the phenomena by which we can basically tell that whether a protein is mobile or immobile in the plasma membrane or some organo-Golgi membrane, mitochondrial membrane. So what you do that you can just use a cell surface protein. Here we are looking at a plasma membrane protein. We can do that many different ways, labeled with fluorescent antibody. They can be expressed as fluorescent protein. And here is or an RFP tag protein. You can use anything. And then you focus it on a particular part, not the entire cell, a particular part very strongly, but with excitation wavelength and with a laser with a particular wavelength. And what happens when you constantly keep that laser on, it's going to bleach the fluorophores out. So you are not going to visualize anything. So what you do is like after that, you just allow the laser, you turn off the laser and you allow that recovery. And then you again turn on the laser for a short period of time and take the data. And then what you can do, you can just turn on the laser on and off for a short period of time. Remember that if you're just turning on the laser to acquire the data, which could be like 1 millisecond on and off, then you are not going to bleach out, but you're going to get the data. But if you just keeping it on for a couple of seconds, or it's going to bleach out. After recovery, you can take those turn on and off to acquire the data. That's how you collect the data. So what happens that if a protein is mobile, then when you have the recovery period, the proteins from the different areas of the plasma membranes will move into the area which is got bleached. Because all the proteins on the plasma membrane surface, if they are not localized into some particular regions of the membrane that we're going to see in the second module methods, most of the proteins are motile and move around, except some which are particularly done. So if they are motile, you are going to get the recovery back. If the proteins where you are basically focusing, if it is a particular area where the proteins are not moving in and out, like a lipid raft area that the details in the module 2, then you are not going to see any mixture. You are going to see that area bleached as well, even after that. That's the idea. So you have the membrane protein and this bleached area. The proteins are just moving just by random Brownian motion. That's a natural thing. You can just get the recovery. So this is how we are going to see some of these questions. They also happen. Here you have the fluorescent intensity, which is the arbitrary, and fluoresce the area of the cell for a long time. They're all bleached out. And after that, so you can see that you have a high fluorescence. And when you're constantly bleaching it, it will drop. So this is basically bleached completely. I mean, it will not go to zero, but it's just significant drop. Now what you do that you will now turn on the laser on and off for a very short period of time, millisecond time, on and off to acquire the data over the period of time. You are not constantly turning on the laser just like this. If you constantly turning it on over the period of time, that you are going to bleach it back again. So these are some of the technical things in there, not going into the detail, but long time in bleaching and then just acquiring the data, which needs just short burst of laser and collection of data. And you can see right here, the 50% of that right here, 3,000 to this, and see that this is about, say, 1,500, so half of it, so of this arbitrary value. So if you just look at an arbitrary value of about 1,500, half of the original value, we will conclude that 50% of the proteins are mobile, 50% immobile. So what kind of question we can ask? So some of the typical question that we can ask, just give you a graph like this, and if it is like straight right here, which is at the base. So here we will say that these are 100% immobile. So it would never recovered So it never went down in there. And theoretically, we can just also say that it just goes all the way up here. And then here, if it goes all the way up here, we'll say that these are 100% mobile, which is hardly with the case. You can have 90%, 80% mobile. But technically, I'm saying that if it is coming back to the same level where it started with, theoretically, this should be 100% mobile. If it's not going anywhere, it will be 100% immobile. If it's somewhere in between, you can see if it is exactly the same value in here. And again, you just visual expression. We will not go like this or this. You do not have to calculate or do any graph. You can just have just only these three types of things. In the middle, 50% mobile, immobile, 100% mobile, or 100% immobile. These are the scenarios that you can just do some data interpretation. After that, we are just going into the FRET. So the FRET, fluorescence resonance energy transfer. So what we can do that we can actually tag different fluorescent protein pairs in such a way that we can study protein interaction. And we are going to study this a lot all throughout the other chapters as well. This is just the first start. We are going to see this same slide over and over in all different sections. So you have to choose a FRET pair in a way that see that first here is a CFP. You have excitation wavelength, emission wavelength. The YFP also will have an excitation wavelength, emission wavelength. So what you choose to do, have a FRET pair for two different proteins. The emission spectra of the first one should be the excitation spectra of the second. So what happens that if these two proteins, which are tagged with, are closer than 10 nanometer, these emission spectra of the one can be transferred to the second one as the excitation spectra, and you are going to see the emission spectra of the second one, of the acceptor. So that's basically the idea. So if you see that there are two proteins in a cell, and you have an emission of cyan fluorescent protein right here, you do not see any FRET. But if you excite them with the excitation of the CFP, cyan fluorescence protein, and if you see an yellow emission out of that, you know that there is interaction between the CFP and YFP, because this is only possible if they are less than 10 nanometer. So to understand the interaction of two different proteins, protein-protein interaction, whether one protein interacts with the other or not. So you can also see the lightboard video. So I just explained that. All right. So super resolution microscopy, really beyond our scope. Universities also do not have that. Just to know that what is out there and possible to do those. So just you need one line. All you need, basic definition, working principle in one sentence. Basically, that's it. So theoretically, computationally, they can push the resolution of theoretical optical microscopic limit beyond that. So 200 nanometer is the theoretical limit of the resolution. With the structured illumination microscopy, you can push it to 100 nanometer. But remember that this is digital enhancement. This is not an optical enhancement. So this collect images illumination in different patterns and generate interference pattern that can computationally reconstruct to create the image. So these are all digitally measured. And now here is also a stimulated emission depletion microscopy where you use an excitation laser point, and around that you put a depletion beam in there. So what happened, the depletion beam is just like your confocal, which is physical entity. Here it's an illuminated entity. So this depletion beam doesn't allow these outside emissions to come through, and this only takes just the middle sharp points in there. So that increases the resolution for that. So usually what we use for that excitation laser point surrounded by a donut-shaped depletion beam. So this word is important. That's why I said that if you just see that, a donut-shaped depletion beam making the area much smaller, what kind of microscopy that use? We will use these words, and you know that it is STED. And it can go up to 30 nanometer resolution. You can see that right here, which looks a little blurry in a point scanning confocal. And you can see that separate out these tiny fibers because depletion beam doesn't allow things. Then you have a photoactivated localization microscopy, which randomly just illuminate different points and then take a Gaussian distribution to see that exact midpoint of that and then they can essentially a finest image because they just subtract all the background images and just take that median point and just construct that image. Photoactivated localization microscopy. Just one sentence. So it's super, super easy. This is a light sheet microscopy. This is actually used for most of the time live specimens. These are used routinely in the zebrafish visualization. So you put a zebra embryo, which is alive, in soft agarose, and it is still alive. And at that time, you have this detection and the illumination objective. So the idea is that you put the illumination and the detective objective in 90 degrees. See that for all of these are just like conventional. You have the source, on top of that, you have the objective. Source, top of the objective. These are the regular ones. Here, you have the objective and the detector in 90 degrees. So what happens for that, see that right here, the illumination is illuminating the specimen, this entire sheet or a plane of the zebrafish from the tail all to the head. And the fluorescence is going in this direction, and that is being detected. So by light sheet, we mean these lighted plane that you are essentially seeing in this process. So that's basically the idea. See that here, the light is wide field or confocal. The light is going up and down. Light is going up and down. Here, light is coming as a sheet from the side, and in 90 degree, it's detected. And this you can just use for the live space visualization. Just remember one important question. There are two of these microscopes that can visualize live specimens. One is this light sheet microscopy. Definitely underline that. And another one is your 2-photon excitation microscopy. These are the only two that can visualize live specimens. See that right here? It's in yellow. That means these will be your exam questions, live specimen visualization. All others, you cannot do it with a live specimen. Very important. So remember that. All right, electron microscopy. These are all the same for electrons. That only the resolution is much higher because electrons, when move in a fast pace, can act as a wave. You can see that the nanometer is 0.005 nanometer wavelength, which is very less, and that's why 0.1 nanometer could be the resolution for that. Light and electron microscopy. So instead of those lenses, you have these magnetic coils, and you visualize your specimen in a three-dimensional microscopy, like, for example, not three-dimensional, in a transmission electron microscopy. You cannot visualize the specimen directly because this is an electron beam which are invisible. It's falling onto those, and then it must fall onto the detector. And this detector, just like your old school television screen, which has phosphor. So the electron beam, when it hit those phosphor detectors, you see a dot which is illuminated when electron is hitting it. If it's dark, then it's not electron. These are like all those box TVs that you used to have, those cathode ray projectors. This is exactly the same way you work. So you have the tungsten filament here, you put your sample, the beam passed through it, and right here at the bottom, you have this phosphor screen, and you can visualize your specimen with this. So this is your transmission electron microscopy. It will pass through the specimen, so your specimen must be thin enough to pass those electron beams. If it is a thick specimen, you cannot do a transmission electron microscopy. To do a thin specimen, how you can do that if it's a thick? You can fix them with glutaraldehyde, osmium tetroxide. Red arrows means that these are important for your exam. Uranyl acetate, osmium tetroxide, remember that, what they do. You fix the specimen, then you embed usually in an epoxy or a solid plastic block. You do it with a wax in your regular microtome. You use plastic or epoxy. Then you section them using an ultramicrotome or cryomicrotome, about 50-100 nanometers thick. Then you do a negative staining. You cannot use a regular colored staining. So see that all these uranyl acetate, lead acetate, osmium tetroxide, phosphotungstic acid, these are all heavy metal salt. So when you do the negative staining, the areas where you have these salt deposited, that will be opaque to the electrons. Electron will not be able to pass through those heavy metals, but the areas where you do not have the stain, it will pass. So organelles pick up stain poorly, electrons can pass. Organelle that pick up stain efficiently, most of it will deflect. very few will, so you will have a dark spot. So this is how it looks like. So here you can see that right here, this is just like a plant cell. You can see the nucleolus, you can see the nucleus, and this is the stem of an onion root cell. So two-dimensional image that you can see. Coronavirus, subcellular component, you can see the endoplasmic reticulums. This is phagocytosis of neutrophil internalizing bacteria, two of them, one right here, one right here. Beautiful transmission electron microscopic images. Now, you have an advantage of that. You can also do immunoelectron microscopy. And here, what you do that you have a primary antibody, just like your fluorescence microscopy, but a secondary antibody you raise the same way, but instead of a fluorophore, you attach a gold. It must be a heavy metal to detect that. So this is the important part. If you just add a fluorophore, it has nothing to do, electrons will not excite a rhodamine or a fluorescent or any fluorophore. It must be a heavy metal. And using that, you can just see the localization of a protein. Interestingly, there is another way that you can do this, and this can be done with the your fluorescence microscopy as well. There is a protein called protein A. This is a protein from Staphylococcus aureus. This is a bacterial protein. Interestingly, this protein A binds to the Fc portion of all different antibodies, all mammalian antibodies. It doesn't matter whether it's a rabbit or chicken or mouse or goat. All antibodies, this protein A binds to the Fc portion. So now what they do, that it is easy to grow Staphylococcus aureus in the culture, and they purify the protein A, and they conjugate the gold with the protein A. And this is your reagent, which is much more cheaper than your secondary antibody conjugated with the gold. All bacterial, here you have to inject it with the animal, maintain the animal, bleed the animal, much more expensive. Growing bacteria, much easier. Purifying the protein from a bacteria, much easier. Conjugate with the gold. So what you do that, you have your primary antibody, which is against a catalase right here. The antigen is catalase. You put your primary antibody. This is your primary antibody. And you add your protein A gold instead of a secondary antibody, and you can use those. So the question is why you need to do the immunoelectron microscopy. So here your organelles stained with the immunogold, and you can see those. So here we are coming here. So suppose you are getting an electron micrograph of this one, and you have these vessels right here. You can see you have 1, 2, 3, 4, 5, 6, 7, couple of these vessels. Now, without immunoelectron microscopy, all of them looks, so this will be empty. And you have no idea, are these peroxisomes? Are these lysosomes? Are these mitochondria? What are they? You're confused, and you cannot tell. So in this case, you can do an immunoelectron microscopy to determine that what exactly are you seeing. As I said, just like the plasma membrane or the cell surface, all the different organelles will also have these marker proteins. So in the peroxisome, you have these catalase is the marker protein for your peroxisomes. So on the other hand, say for example, if it's a lysosome, say for example, acid hydrolases like an hexokinase or acid hydrolases will be the marker protein. So mitochondria will have their own marker protein. So peroxisomes, catalase is your marker protein. So you conjugate the catalase with the primary anti-catalase primary antibody, conjugate with the protein A gold, or a secondary antibody against the primary antibody. And now you see that these dots appear in these two, but not these. So these are two peroxisomes, but these are not. They are some other organelles. So you can just pinpoint what they are. That's the objective. All right, so you can do cryo-electron microscopy, which you basically use your transmission electron microscopy, but the sample prep and imaging technique is different. First, you put your sample in water, which is vitreous ice. So, which is amorphous ice, remember that. So basically you take your sample, whether it's a cell or virus or anything like that, and you dunk that in liquid ethane. So it minus 150 degrees Celsius, it instantly freezes in a vitreous form, doesn't form crystalline ice. The advantage is that crystalline ice with these jagged ends that destroy any biological structure. But when you have an amorphous or vitreous ice, it's like glass-like. It will not destroy and preserve the exact three-dimensional nature of the specimen. Now you have the sample and you shoot the electron beams to that and you tilt the image plus minus 70 degree, plus 70 degree to minus 70 degree in 1 to 2 degree increments. And you take these sample images in little bit different angles and you collect those and computationally you reconstruct with Fourier transform these images. So these are again multi-million-dollar instruments. You can get amazing details. When you are reconstructing an image, suppose if you're just looking at a virus with the cryo-EM, it's just looking at the virus. It is not reconstructing image. But if you are just when reconstructing an image, this is just like a reconstructing your MRI image or a CAT scan, computer-aided tomography. That is exactly the same thing. You just take the images and here you have the nuclear pore, or you can see that the different planes of that, and the it can reveal the 3D tomographic image near the atomic resolution as well. So earlier, only X-ray crystallography used to be used. Now this cryo-electron microscopy is taken over. It's much more straightforward, and the computational power has gone through the roof in the last 20 years, and you can just get these amazing details. Scanning electron microscopy, on the other hand, can give you a 3D image reconstruction from a 2D, and the ACM gives you a 3D image resolution lower than 10. See, remember that cryo-electron microscopy provides two-dimensional information. 3D images are reconstructed from 2D. Here, the collected images are two-dimensional. 3D construction is through the computation, whereas ACM provide actually a 3D image resolution, but it's lower than TEM. But here, you can use a whole cell or unsection tissue specimen that are fixed, dried, and must be coated with a heavy metal, such as platinum or gold. You do not coat a sample for TEM for any of those. You either stain them, which partially will put the negative stain somewhere, or you just immunologically stain them, but you do not cover the entire cell. Here, what you do, the ACM, you have the electron beam falls onto the specimen, which is entirely coated with the metal, and it will deflect the electron beams into the detector, depending upon the depth of the material. And you can just detect that and reconstruct the image. You cannot visually see it just like the TEM. You can see the images on a phosphor screen with your binocular vision. Right here, you can see them. You cannot see that. It can be only detected, the electron beams, by the detector, and you can only see it in the computer screen. So here you can see that what we are seeing. Nomarsky light, this is the stereocell's cilia from the hair of a bullfrog in your ear. TEM just gives you a 2D information. Here, ACM, you can see that these are the 3D information. See that these tiny little things, can you tell what are those? Tiny little structures, the inner ear. Those are coccus round bacteria from there. Here you can see that these are some of the cilia from the cells. These are lower respiratory cells. Goblet cells are secretory cells. So they have made a section of these, then completely covered it with the metal, platinum or gold, and then take the three-dimensional image with that. You will not get this kind of picture of the cells without the ACM. If you take a picture with the TEM, it will be looking like this, two-dimensional, whereas you can get these kind of pictures with the ACM. Yep, that will be it. All right, last part. So let's take a look at the whole cell, that we need to separate the organelles depending upon that what we need to do with all different purpose, what you exactly do. So you take these whole cells, which can be from the tissues or the cells you are growing, and then you disrupt or break them up by sonication, tissue homogenizer, blender, or force through small pores or a filter. Sometimes we use the syringe filters to pass the cells through those, and the pressure will just break them. So after that, we need to centrifuge them. But when we usually do this process, we need to always keep them on ice during the cold. And you have to add these PMSF or other protease inhibitors. Remember that we talked about that. We'll come back to the drugs used in the cell biology. This is the one which was at the very top of that list. So what is the reason for that? Remember that you have all these lysosomes in the cell. When you break the cells apart, these lysosomes can break as well. And they have all these lipid, protein, carbohydrate, nucleic acid digesting enzymes. These are all acid hydrolases, and they can digest the content of the cell, but it can do that the same thing if are not careful. So ice, first of all, keep the enzymes not working. So that's one of the inhibitor, of course, for all the enzymes in the cells. And when you add the protease inhibitor, these protease, most of the components in the cell, most of them are proteins. Of course, there is carbohydrate, lipids, and other, but the proteins are the ones which basically give the structure that you need to worry about. And you use these protease inhibitors so that proteases do not digest your protein. So that's the important part. Remember that. So once you extract or make that homogenate, you then do a differential velocity centrifugation. So what you will do, that you will take these and pass it through like a crude filter, and the filtered homogenate, you just spin it at 600 G for 10 minutes. You will put the nuclei as a pellet. Then you take the supernatant and then you put it 15,000 G, 15 minutes. You will pellet mitochondria, chloroplast, lysosome, peroxisome, all of them in the same. Take it, 100,000 G, plasma membrane this, pour out, then ribosomal subunit, and then you have a soluble part of the fraction. So you can just do this. Now here, what you have, there are two things that is needed. Suppose you want to isolate a mitochondria. They are all mixed in here, so you have a problem. You have to do some additional thing that we are going to see that how you do this with the equilibrium density centrifugation. But the second conceptual question that we ask is, suppose we have filtered this and we skip that 600 G right here and took the filtered and directly spun it out at 15,000 G for 5 minutes. Make sure we skip that 600 G directly. So what you will have in the pellet, remember that. You will have your mitochondria, chloroplast, lysosome, peroxisome, and what else will you have? See, the nuclei was pelleting at 600 G. You did not do that. If you skip that, the nuclei will be pelleted at 15,000 G right here. On the same way, if you think about that, you just take your sample and do 100,000 G centrifugation for 60 minutes. You will have plasma membrane, microsomal fraction, fragments of endoplasmic reticulum, and large polyribosomes. Along with that, if you have skipped these part, you will have mitochondria, chloroplast, lysosome, peroxisome, and nuclei. So here is a question that we can ask. Suppose your research only need the soluble part of the cytosol. How you are going to prepare your sample? All you have to do, you take your sample and directly put in a 300,000 G in 2 hours. Every cellular organelle will precipitate in there because you do not want them. You only need that. So you can modify that technique. You do not have to go step by step. If you purify a certain thing in certain step, yes, you do. But if you do not need or care for the other things that is in there, you can just directly skip that, particularly for a soluble fraction right here. Suppose you can just see that right here, you have plasma membrane fraction right here. Then you can easily skip this part and do a 15,000 G. All of them will separate. So you save 10 minutes of centrifugation time. You separate those. And of course, you get these plasma membrane, these fractions in here, and you will separate them out. So these are some of the conceptual questions. So remember that if you skip a step in the velocity centrifugation, in the next step, that previous step's material is going to precipitate. That is your, all right. So after that, what you do, that suppose you take these mitochondria, chloroplast, lysosome, all mixed together, and you do an equilibrium density gradient centrifugation. These are usually plastic tubes, and it can be punctured by sharp needles, which fit into the syringe. And you make all these gradients, which can be made of sucrose, phycol, cesium chloride, many different types of gradient material. And you just make these solutions, and you can take a 1 ml micropipette and just gently pour each layer on top of each other. Or there are some peristaltic pump, which are gradient maker. They will automatically make the gradient for you. You just put a tube in there, and they're just going to just make this gradient automatically by mixing two solutions together. So anyway, so then what you do, that you gently put your organelle fraction on the top, and it's going to float on dust. This is because this is heavy. This is much more denser. It's not going to mix immediately. It's going to float. Now you centrifuge them. And what's going to happen that this organelle fraction is going to move through the gradient and it's going to get maintained during the centrifugation. And they will just stop moving at the density where they equal to the density of that organelle. So here, see that peroxisome will go to all the way to 1.23. Mitochondria will stop at 1.18. Lysosome will stop in there. So how you will collect that? What you will do that you will puncture the bottom of the tube and you will collect the fraction, and you will take the highest density at the bottom, collect it first, and you collect the fraction. So that's how you will purify that. So how you would know where is your sample? So suppose you are purifying your mitochondria. Using your mitochondria that was somewhere here. How will you know that where is your mitochondria? Again, remember that we said that all of them do have some marker proteins or enzyme. You just plot that these are your fractions. So you have your 1, 2, 3, 4, 5, 6, 7. And if suppose you just collect 100 microliter in each one of them, you just add 100 microliter of fraction. Then from the 100 microliter, you can just take, suppose 10 microliter of your sample, and you just do an assay. Usually these are enzymatic assay. You just measure an enzyme activity for a mitochondrial protein. So there are several mitochondrial proteins which are only available in the mitochondria, which will not be available in the lysosome, which will not be available in the peroxisome. Peroxisome, you can use the assay the catalase. The same way you assay that. And then you plot the activity of that enzyme on these. Suppose you get a curve like this. This is your enzymatic assay activity. So you know that your fraction 4, 5, this has the highest activity of that protein. So you collect those 4, 5. That will be your fairly pure sample of your mitochondria. So this is how you collect those fraction. And now if you want to do your experiment, cell biology experiment using your mitochondria, you can do those experiments. All right. So after that, it was just last 2 slides, and it will be super, super easy that how you can detect the proteins in a cell. Literally, this proteomics is now that such that you can take an entire organelle like a mitochondria or peroxisome and know each and every protein that you have in there. And this is usually done by mass spectrometry. There are many different TOF. This is a multi-TOF. It's known as matrix-assisted laser desorption ionization time of flight. So this is basically the matrix, which is the metal target. And you put the protein samples on there, and it is hit by a laser, and it is ionized. So these fragments of the peptides are going to get fragmented, and there could be small and large fragments, and they are going to move towards a detector which is negatively charged. And these are plus charge. These tiny pieces of fragments, they are going to move. And the flight of detection, the lightest ion arrive at the detector first, heavier later, and you get these peaks. And you computationally can find out what is the time of flight and what could be the peptide sequence of that fragment. It's amazing computation that you can just find those things out. And by that, you can tell that what are the different fragments, put back them to just not only one protein, but hundreds and thousands of proteins. And you can tell that these were the proteins and these were the fragments. It's amazing kind of technology. So how you do this? What proteins are found in the mouse mitochondria? You do the tissue homogenate, that right from there, differential and density gradient centrifugation. You have to do both to purify the mitochondria. And you digest the protein by the trypsin. This will digest them into small peptides. Remember that. All the proteins, hundreds of different proteins, small tryptic protein. These are your tryptic peptides. Now you add them, adsorb onto the matrix. Remember that it is the MALDI-TOF, which is the matrix assisted. This is your matrix. You put the sample in there. You hit it with a laser. Now these tryptic peptides are going to get ionized and go depending upon your small and larger size. And you can find out that which are the peptides, what are the different sizes, and you can find out the amino acid sequence from there, compute back what were the peptides, what were the proteins compared to the database to the ID proteins. So just an overview of that, how you do this, and that is more than enough for that.